Rajesh sir, hello. Sir,
Yes.
we are live now.
Madam, you are muted.
Yeah. Somewhere something incredible is waiting to happen. Carl Saxe. And the same way, somewhere incredible is already happening, and that is our FDP. Good evening, ladies and gentlemen, and all my dear participants and speakers for the day. It gives me immense pleasure to welcome you on the sixth day of our FDP. This FDP, which is organized by International Journal of Microbial Science in collaboration with Sushila Shankarrao Gadhave Mahavidyalaya, Khandala district Satara. We almost have finished our 5 days, and today is our sixth day. Looking forward to yet another and different topics of statistics. And the role of statistics in our academics and research we have been learning through all these days. So, with a great zeal of learning, we are looking forward for today's presentations and for the speakers to be heard. Without much ado, let us begin our technical session for today. Before we move on, I would like to make a notification here that participants are requested to post your attendance on YouTube, and at the same time, do write your MCQ test on November 8th, 2020, because that will be the one criteria for you all to be certified for this FDP, which is immensely required for your API score as well. With this, we will proceed to our first session of the day. The first session of the day is on the topic regression coefficient, error bars and graphs for figures, Pearson's method for determining correlation coefficient. And the speaker is Dr. Ashok Bankar, Department of Microbiology, Abha Sahab, Garware College, Pune. I request Minal Majati to introduce our speaker to the participants. Over to you, Minal.
Madam. Good evening, everyone. I extend a warm welcome to all on Faculty Development Program. It is a great honor for me to introduce and welcome our speaker of this evening, Dr. Ashok Bankar, sir. Dr. Ashok Bankar, sir, is currently working as the assistant professor at Department of Microbiology, MES, Abasaheb Garware College, Pune. Sir completed his MSc in microbiology from Savitribai Phule Pune University, Pune, and PhD from Institute of Bioinformatics and Biotechnology, Savitribai Phule Pune University, Pune. He has more than 18 years of research and teaching experience. Sir received 9 foreign travel grants award from different agencies like CSIR, DST, DBT, European Science Foundations, and BCUD, Savitribai Phule Pune University, Pune. Sir presented his research work in various state, national, and international conferences and seminars. About 46 research papers published in a reputed national and international journals by Sir. Sir is currently working on a major project on application of bio-inspired nanoparticles in improvement of plant crop productivity, an interdisciplinary approach of 39 lakh 42,500 rupees, funded by DST SERB, New Delhi. Dr. Bankar sir is a reviewer of 23 national and international journals, as well as he is a member of editorial board of Journal of Nano and interdisciplinary research. Was organizing committee member for 3 conferences which were sponsored UGC. Dr. Bankar sir was selected for expert scholarship visiting research to Göttingen University, Germany. Sir was invited as judge as well as a guest speaker for many programs. as Academic Research Coordinator and NAG Steering Committee Coordinator for Criteria 3rd. Sir also worked as 4 times observer for MH SET examination. Sir guided many MSc, MTech and PhD students for their research projects. With this introduction, now I hand over this session to you, sir. Thank you.
Thank you, Amal sir, for such a nice introduction about me. So I hope you can hear me nicely. So I welcome all the participants for 1 week faculty development program on statistical analysis in academics and research. So my today's topic is regression, correlation and error bars in graph. So how this regression, correlation and error bars are important in academics as well as in research. I'm going to explain all the importance of this topic. So as sir told that I'm Dr. Ashok Bankar working as assistant professor in Department of Microbiology, MES Abasaheb Garware College. And this 1 week faculty development program is organized by International Journal of Microbial Science in collaboration with Sushila, Shankarrao, Gadhave, Mahavidyalaya, Khandala, Satara. So already sir explained about my achievements and all that things. So here you can see that my citation index, H index and I10 index. So you'll understand. So let's start with regression analysis. So first thing is that what is a regression analysis? What is the importance of this regression analysis in the statistics or in research or in academic? So first thing is that regression analysis is a statistical tool which is used widely in academics as well as in research work. So in regression analysis, so the regression line plays very important role. So first we need to understand what is a regression line. So this is a regression line. So what is this regression line? So it is a single line. You can see that it is a single line that best fits the data. Okay, so in graph here, I showed that how this single line is well fitted in the data. So what is the data? So data is nothing but whatever the information you have collected. It may be the microbial cells, it may be the plant flowers, plant leaves, seeds, anything in the particular area or depending upon what kind of study you want to do. So just you want to find out is there is any correlation, is there is any relationship between the two variables. Okay, so this is a data. Here you can see that this line is well fitted. So why it is called as well fitted? Because all these points are around this line. Okay, so they are not very much deviated. So this is a normal distribution. So whatever all these points are near to this line, that's why this line is well fitted to the data. And that's why it is called as a regression line, because it is well fitted. If these points are here, some points are there, so some points are at below, some points are there, that means the data is not well fitted in the single line. But here you can see that now the data is well fitted, and that's why it is called as a regression line. Now first we need to understand there are two variables. For example, variable x and variable y. So there will be two regression lines. Okay, suppose for example, if you are considering two variables x and y, so you will get the two regression lines. So first line, that is a regression line of y on x, or second line, regression line of x on y. Okay, either y on x and another one is a x on y. And also you will get the two regression equations also. So here you can see that you will get the two regression equations, y on x and another one is a x on y. So these equations are called as a regression equation of y on x, and another one is a regression equation of x on y. So here you can see that the y is equal to a plus bx, xe, that is equation, is equal to a plus by. So here y and here is x. So now in these two these variables, now you understood that so one is dependent and another one is independent. In this case, when you plot regression equation y on x, at that time here y is a dependent variable. So why y is a dependent variable? Because if you do or some changes in x, automatically the value of y is going to be changed. That means small change in the x is going to affect the value of y. That's why y is a dependent variable and here x is a independent variable. In another equation, x on y. So in this case, x is a dependent variable and y is a independent variable. So if you make change in the y, automatically the value of x going to be changed. So in this way, you understood that what you mean by the dependent variable and what you mean by the independent variable. Then you saw in this equation there is a and b. Then what is this a and b? So this a and b are nothing but these are two unknown constant. They are called as a unknown constant. So what is role of this unknown constant? Number one, that is a. So a indicate value of a indicate that how much data is well fitted in that line. Okay, so fitted line is indicated by a, and b is nothing but the slope of line, that is a regression coefficient. So b, slope of line, and a is a how line is well fitted. Okay, that is an indication of this a and b. Now the next question is that what is the difference between regression and correlation? Okay, so we have to understand that. So regression and coefficient, there is a difference. So in case of the correlation coefficient, it shows the degree of relationship. Okay, it will show the degree of relationship. That means how much one a and b, how it is that they are affecting on each other. But in case of the regression analysis, it shows the nature of relationship. What kind of relationship it is, whether it is a negative relationship or it is a positive relationship. That kind of the relationship is a I mean explained by the regression analysis. And that's why regression analysis as well as the correlation coefficient, both are valuable and both are important. So now we'll understand the regression coefficient. Okay, so what is this regression coefficient? So regression coefficient is nothing but it is a statistical measure of the average functional relationship between one or more variables. Okay, so if you want to study or you want to measure the relationship between or functional relationship between the two variables, so it is not only possible to measure the relationship between the two variables, but you can take more than two variables. For example, if you want to check the effect of antibiotics on the microbial growth. Okay, so this is a two variables. One is antibiotic and another one is a microorganisms. But if you want to check the another one, so you can take two antibiotics. So you are going to check the two antibiotics on the microorganism. So there are more than two variables. If you want to check the effect of temperature on the growth of microorganism, if you want to check the effect of pH, temperature, as well as the other factors. So like that, more than two factors or more than two variables, we can study it in the regression coefficient. So what are the properties of regression coefficients? So simply it is denoted by B. As I said in that constant A and B, B is nothing but it is a slope of line, and that slope of line is nothing but it is a regression coefficient, which is denoted by B. And it is also expressed in terms of original unit of data. Okay, so data is expressed in terms of the original unit. So what do you mean by the original unit? For example, if you want to study the different soil samples, okay, and that unit is in kg, or that unit is in gram, or that unit is in the kilogram or mg, or simply gram. So you can express the same unit, whatever the original unit you are using, that same unit. For example, water sample analysis. So you can use the unit liter. So like that, it can be expressed in terms of the original unit of data. So between two variables like the x and y, two values of regression coefficient can be obtained. We can easily obtain the two values of regression coefficient, and both regression coefficient must have the same sign. Either it should have positive or it should have negative, and any sign, but both will have the same sign. So regression coefficient are classified into the three main groups, that is simple, partial, multiple. Another one is a positive and negative, and third one is a linear and non-linear. Okay, so then the regression coefficient can be worked out for the unreplicated data like this. So this is the equation for the unreplicated data, that is a byx, that y on x and x on y. So this is a equation for the unreplicated data. So what do you mean by the unreplicated data? So unreplicated data means that data we cannot replicate for the several time. For example, what is the heavy metal concentration? What is the heavy metal concentration of particular concentration in river in winter? So same heavy metal concentration will be changed in summer, again it will be changed in spring. So like that seasonable, there is a variation in the heavy metal concentration. So if you go for the replication or you want to analysis, you will get the variation in your heavy metal concentration. So that means this data cannot be replicated. Suppose you took the sample and now you got the 200 mg per liter. So it is not guarantee again you will get the 200 mg per liter in the next season, because the composition of water is going to be changed. Such kinds of the data is called as a unreplicated data. For example, in election, suppose X person got 900 votes, and again election is happen. It is not guarantee again that person will get the 900. Maybe that person will get more or that person will get the less, and such kinds of the data is called as a unreplicated data. For example, in cricket, for example, Sachin Tendulkar is playing, and in this match, if he got 100 runs, it is not guaranteed that again in second match he will get the 100. Either he'll get more than 100 or less than 100. Such kinds of the data is called as a replicated data. Another data is called as a replicated data. So replicated data means there is a guarantee that again and again you will get the similar kind of results. Okay. So such kinds of the data is called as a replicated data. For example, if I am writing any research paper and I have data, and suppose the reviewer or editor have some doubt that they want to check whether if I am saying that reaction A plus B is giving D, so this is a replicated data. That means when I am adding A plus B, I should get D only. If I am getting E, that means the data is a faulty, data is a problematic. So if the reviewer or if the editor want to check, is it real A plus B is equal to D, then they will check it. And if they are got A plus B is equal to D, that means your data is valid. Your data is replicated, and your data is have more value, because the whatever the experiment they have carried out, the same result they have obtained. Such kinds of the data is called as a replicated data, and such a replicated data also can be obtained with the help of the following equations. And in these equations in this equation, then you can put all the values and you can find y1 x as well as the x1 y. So both the regression coefficient can be obtained for the both the variables, and the value of the both the variables will be, a sign will be the same, either positive or the negative. Now the next thing is that whatever the you got the data and regression coefficient, you got it, and still people want that whether the your data, whatever the your analysis is correct or no. If they have some doubt in their mind, again we have to take the help of the another test, and that is the t-test. So here you can apply the t-test for the further queries or further clear all the doubts. So in this way, this regression coefficient have several applications in scientific studies, as well as the in medical science, in biological and agriculture science, and also to understand the effect of relationship between two or more plant characters, as well as to predicting the performance of selected plants in the next generation. Whether your next generation of this particular plant will be useful or beneficial or productive, that can be predicted with the help of this regression coefficient. That means we can understand what is the factor which is affecting on the plant character, on the plant growth, which are the factor that is affecting on the health of the person, that can be studied in the medical science, and which are the biological factors, or the in agriculture, what is the effect of manure particular or effect of microorganism on the growth of plant, that can be also studied with the help of regression coefficient. So these are the different values. So here we'll see one example, how to calculate the regression coefficient and how to obtain the lines of regression for the following data. So here you can see that this data is given x and y, and these are the values of these variables. And then we have to solve this problem. We have to calculate the regression coefficient. So for that, what we have to do, we have to arrange x and y. Then we have to find out the x square. So you'll get all this value in the column. Then y square, you'll get all the values in this column, and y square also. So you'll get xy, sorry, x square, y square, and xy. Okay, then you have to take the summation of x, summation of y, summation of x square. You will get the value here. Then summation of y square and summation of xy. Now you obtain all the values. Then you can find out the mean of x and as a mean of the y by taking the summation of x divided by n. N is nothing but it is a number of population. How many items are there? How much population size? And here you can see that it is
Hello, sir. Are you there? Excuse me, sir. I think we have lost the connection with the sir. He will be joining us within few minutes. Maybe there is some technical glitches over here. I request all the participants to bear with us. He will be joining with us very soon. Yeah, he is on board now. He will be joining with us very shortly. I regret the inconvenience caused to all the participants here. Please be along with us. Hello, Bankar sir.
some net issue.
Yes,
Yes,
we
madam.
understand there will be some lags here in the technical section. You can carry on, sir. Your slides are also visible and you are audible as well.
Okay, thank you, madam. So I was talking about this problem, that is how to calculate the regression coefficient. Then you plot.
Sir, could you go back, sir? Actually, you lost, your connection was lost.
Then you have to take the values of x from regression coefficient classification here.
Yes, sir. This slide was
From
visible
this,
during that time. If you have
okay.
technical lags,
So from
can I
this,
share your screen, sir? Can I share the PPT?
yeah, you can share. Hello, can you hear me now?
Yes, sir. You are audible now, sir.
Okay. So a regression coefficients here I will explain what is a regression coefficient. So it is a statistical measure of the average functional relationship between the two or more variables. Okay.
I think we lost him again. He will be joining us in a short while. I request the participants to stay tuned with us. And I apologize for the technical snags what are happening. So as you all know, there are always pros and cons for each thing. I think online virtual meetings this is the only cons which we can say, the technical issues which we don't know when it will appear. So I request all the participants to bear
So
with us.
can you see, madam, my presentation?
No, sir, your video is visible. Presentation is not visible.
Just a minute.
Meantime, I would make an announcement that participants who have registered but not have applied for the certification can definitely pay a minimum amount for the certification after you clear your MCQ test, so that you will be getting certificates for your FDP. Hopefully, everybody is doing. I think because during this pandemic time, everybody is putting all the efforts to stay in tune with what is happening in and around the world, and we are trying to do that with all our best. And because this 4th to 6th of November is International Stress Management Day or Stress Day, so hopefully everybody will try and strive to be free of stress. Yeah, we have him again on the boat. Hopefully he will be starting soon. Can you resume, sir?
Yes, yes, madam.
Yes, sir.
So I was talking about the regression coefficient. So it is a statistical measure of the average functional relationship between the two or more variables. I was talking about the two or more variables. That means antibiotic and their effect on the microorganisms. So there may be two or more antibiotics. Okay. So we can also understand the effect the temperature, pH, as well as the other factors on the growth of microorganisms, and we can understand what kind of the relationship, whether it is a toxic relationship or is a useful relationship. For example, amino acids. If there are the amino acids, these amino acids helping the microorganism, so our microorganism will grow. Okay, so such kinds of the functional relationship can be easily understand. So properties of regression coefficient, as I was explaining, so it is denoted by the B. So B here I already told that B is nothing but these are the unknown constant, and it is a slope of line, that is a regression coefficient.
Sir, for your convenience, can I share the PPT on my screen, sir?
Yes, madam, you can share, please.
Yeah, I think this slide we have finished just now, no, sir?
Yes.
Sir, you please stop sharing screen, sir, so that I can do that. I'm not allowed to do it. Am I audible, sir? Can you please stop sharing so that I can proceed with sharing, sir? I think we have lost him again. Maybe he will be resuming soon along with us on this platform. Until he comes back, I'll be sharing some information with you people here. So, as we are all discussing about analysis on research and in academics, actually there are some important things like API scoring and all. So, according to the table 3A, criteria for shortlisting of candidates for.
Madam,
Yes, sir. Are you back, sir?
madam, you please share it.
Yes, sir. You can stop sharing. I'll start sharing my screen, sir.
I already stop because I can see that.
Okay, sir. Is it visible now, sir? I have shared the screen,
Yeah,
sir.
percentage just, yeah, just reduce the percentage, madam.
Okay, sir.
Okay.
So this is slide number 6, which I have shared here, sir. Is
Okay.
this okay?
So, okay, madam. Uh, 85. Okay.
Yes, sir.
So, as I said, the regression coefficient that can be easily we can easily classified it, and there are the groups of the regression coefficient. That is a simple, partial, multiple. This is one group. Positive and negative. This is non-linear. So the then regression coefficient can be unreplicated data. So what do you mean by the unreplicated data? So that means in that data unreplicated data. So what do you mean by the unreplicated data? The data that cannot be replicated or that cannot be produced continuously. So such kinds of the data can be also studied with the help of the regression coefficient. So these are the regression coefficient equations are there. Byx, that is y on x, and then x on y. So unreplicated data, for example, the heavy metal concentration in the particular river. And if you go in winter and if you go in summer as well as in the rainy season, at that time the same concentration will not be there. Okay, so if you go to find out what is the heavy metal concentration in the summer or what is the heavy metal concentration in the winter, at the same time you'll not get that same concentration. And such kinds of the data is called as a unreplicated data. For example, Sachin Tendulkar played in particular game and he scored the century. It is not guaranteed that again in another match he will get the 100 runs. So that is such kinds of the data is called as the unreplicated data. And the replicated data, so that means in that data you can reproduce the your data. For example, if I am saying that the reaction A plus B give the product D, so even inward anyone when perform this reaction A plus B, at that time there will be the product D only. So that means our data is a replicated. That means authentic data is a authentic. That can also be analyzed with the help of the regression coefficient. That is with the help of these two equations, that is a byx and the bxy. That means y on x line and the x on y equation will get it. Next line, next slide.
Yes, sir.
So this regression coefficient, they have the different applications in the different fields. For example, they are useful in the scientific studies. For example, if you do study of the particular, for example, effect of cigarettes on the health of human being, or the effect of the particular viruses and their interaction with the other viruses or other microorganisms, or association between the two microorganisms, association between the two plants and microorganisms, association between the human being and the microorganism. Such kinds of the scientific information, scientific data can be collected and can be analyzed with the help of the regression coefficient. Then in case of the medical science, okay, so effect of the particular virus on the health of human being. For example, effect of coronavirus on the health of human being, effect of coronavirus on the respiratory tract system, effect of coronavirus on the kidney or on the brain. Such kinds of the study can be done with the help of the regression coefficient in the medical science. Similarly, in biological and agriculture science also, we can find out the effect of manure or particular microorganism and their growth on the their effect on the growth of plants, plant productivity, that can be also studied. Then another one is the effect of the
Hello, sir. Hello, sir. I think we have. Sir, please unmute yourself, sir. Sir, please unmute yourself, sir. You are not audible. Dr. Bankar, sir, can you please unmute yourself?
just a minute.
Yes, sir.
Can you hear me?
Yes, sir.
Just a minute, madam. Next slide, please, madam.
Yes, sir. Is this right, sir?
Next slide. Okay.
Yes, sir. You can carry on.
Okay. So here we can find out the madam, back, just go back to your, you just
Number 8, sir.
go back, applications. Yeah. So I was talking about the applications of this regression coefficient. That means in scientific studies in medical science, as I told, that the effect of coronavirus on the health of the human being, effect of the coronavirus on the kidney, effect of coronavirus on the brain, that can be studied with the help of the regression coefficient. In biological and agriculture science, that is also we can study it. The effect of relationship between two or more plant characters, that can be also studied with the help of the regression coefficient. So we can also predict the performance of the selected plants in the next generations. That means what is the effect of the particular plants on the next generation, how that plant is going to behave or how the productivity will be there. So we'll see the in next slide how the we can apply this regression coefficient. We can calculate the regression coefficient and obtain the lines of regression for the following data. For example, here you can see that the data x and y. Okay, so x and y are the variables. X and y variables are given, and here you can see that the we can plot the value of x. We can also put the value of y, and then we can take the summation of x, summation of y. Then we can also find out the x square value, y square value, and their summation, and then xy value and their summation. Once you calculate that, the next slide, in next slide we can see that how to calculate. So here this is a x bar is there, and this is a y bar. That means it is a mean of x. Okay, that is calculated by summation of x divided by n. N is nothing but the number of total population or number of the items that is selected, that is a 7, and the summation of x, that is a 28. So here x bar is a 4 and y bar is a 11. So regression coefficient of x on y, that we can calculate with help of this formula. Okay, and then in this formula you have to put all the values that we already calculated in this table. Okay, and then after that, whatever the value now you got, that is a 0.929. So that is a regression coefficient. That means your data 92.9 percentage, it is a more relevant, or there is a more relationship between x and y. That is, we are confident 92.9 percent. Then we can also obtain the regression equation of x on y. Okay, so for that we have to just put all these values of, that is a x bar. Okay, so we have to put this value in this formula, and once you got the value of x here, that is a 4, you can put here. Then b xy, that value is got, that is 0.929, you have to put here, and the y minus y bar is already 11. So like that you got the this equation, and final equation, the regression equation of x on y is a x is equal to 0.929y minus 6.219. Here you can see that the x is a dependent, y is a independent variable. If you put, if you change the value of y, that means if you make some small changes in the y, it is going to affect the value of x. Okay, so that's why x is here dependent and y is a independent variable, and thus we can obtain such kinds of the regression equations also. Next slide. Next slide, madam. So similarly, we can also obtain the regression coefficient of y on x, and we can also find out the regression equation y on x. In similar fashion, only there will be little bit changes, and we have to put all the values in this formula, and you will get the value of b by x, that is a 0.929. You have to calculate it. Then regression equation of y on x, then you can also find out the value of y, and in this way you have to put all the values, and you'll get the equation, that is y is equal to 0.929x plus 7.284. So if you make the small changes in this y, x, automatically it is going to the value of y. That means small change in x going to affect the value of y. That means here y is a dependent variable and x is a independent variable. So here in next slide we'll see the for example the particular how to do in Excel. So to calculate the regression coefficient, we can use the different computer softwares such as SAS, SPSS, Minitab, Excel. So depending upon your requirement, um, you can find out different softwares also, and you can have hands-on practice on that software, and you can calculate the values of that particular regression coefficient, or your whatever the problem you have. Okay, either it is academic or it is in the research. So here we'll see the one problem here. So here we are students are given 1 to 10, and these are the scores are there, different marks are there, and this is the IQ of the students and the study hours of the student. Now, in this problem, we have to find out that so there are three variables. One is a test scores, IQ, and study hours. Now we have to find out whether this test score is affected by IQ and study hours. Okay, so that means test score is nothing but it is a uh, dependent variables, and IQ and study hours are nothing but the independent variable. Next. Uh, uh, so next slide, huh? So in this, we have to predict that. We have to find out that whether the what kind of it is a relationship. Whether the really these are independent variable, that is IQ and the study hours are very independent variable or not. Okay, and then we have to predict based on the software. So you can use it in Excel. So best way we can use Excel as well as the any other softwares which is useful or available in your institute or at your research place. Next slide, madam. Um, Madam, I can share now my PPT. That will be more beneficial to me.
Okay, sir. I'll stop sharing.
You stop sharing. I will. Okay, can you see my PPT right now?
Yes, sir. It's
Yeah.
visible.
Thank you, madam. Uh, So now we have to do in Excel. So I just show you how to do all these things in the Excel. You can also open your laptop, and with the help of your laptop also you can do all these things with me also, hands-on practice like that. So you'll understand. So once you open the Excel, so data analysis part is there. So this data analysis, you have to click on this data analysis, and thereafter now as I said, this is our test scores. So 100, 95, all these are the marks, IQ and the study hours. Now click on the data analysis, so you will get the different option, different analysis tool. Just click on the regression and just say okay. Once you said okay, then automatically you will get the you have to give the input of y and x. So mostly on y you will give your, that is a dependent variable. So what is a dependent variable? Dependent variable is nothing but it is a score, and independent variable, that will be IQ as well as our study hours. So once you give the data and just say okay, you will get this analysis. So now here you can see that the intercept, that is a 23.156, IQ 0. That is a coefficient, you got the 0.509, study hour 0.469. Okay, so we got this coefficient. So here we can see that the regression intercept, that is a B, is 23.156. Okay, so intercept, that is a B, is 23.156, that is a slope. The regression coefficient for IQ, that is a B1, that is a 0.509, and the regression coefficient for study hour, it was a 0.467. So what will happen if you put this all value in the equation? So you'll get the equation y is equal to 23.156 plus 0.505 multiplied by IQ plus 0.467, that is the study hours. So this is the only relation linear equation that satisfy our least square criteria. That means this equation fits the data from which was created better. Okay, so our data is well fitted in this linear relationship. So how you will come to know that it is well fitted? Because in the summary you got the R value, that is a 0.905. That means the 90.5% our test score variation, our data is valid. Okay, so that means it is showing that there is a variation 90.5%. That means depending upon IQ, if IQ is less, automatically score is reduced, and study hour is less, study that means score is low. So IQ as well as study hours is going to affect significantly on the scores of the students. That means scores which is a score or marks obtained by the students are because of their IQ and the study hours spent. So this is going to affect significantly. That's why there is a 90.5% variation in the test scores. That means our marks are affected by IQ as well as the time spent for this. So in this way, I will also show you the video. So how you can do it in the Excel.
Now practically
You
here
can do
we
in
will
Excel
see
by
how to use Excel for the analysis of that problem that already we have studied. So first we have to click on the file. After clicking on the file, then you have to search for the options. So click on more, and there will be the options. Click on options. After clicking on the options, then you have to click on the add-in. Okay. So after clicking on the add-in, there is a tool that is the analysis toolpad. So you have to click on that, and then you have to say here go. Okay, after go, then you can select the different options are there for analysis toolpath analysis and all that. Then after that, you have to say okay. So once you did okay, then click on the data, and at the right side here you can see the data analysis. Okay, so click on data analysis. After clicking on the data analysis, there will be different analysis tool, that is ANOVA, single factor, ANOVA two factor, and there are many more tools. So from these tools, now we are going to choose the regression. Okay, so click on the regression, then say okay. After okay, then if you want new sheet, you can click on the new sheet, or if you want the output range, then just click on this and just click here. So you'll get the result here only. So first input y. So this is, for example, y as a dependent factor. So click on this. So this is your dependent factor, that is a total score. But if you want labels, so you have to choose like this, total score. Then on the x-axis, we take the IQ and the study hours. After that, so here you got the summary. Okay, so now you can see that the R-square value, that is 0.9, we got in that summary. Also here you can see the regression and all the data is there. Intercept also we got the 23.15, IQ coefficient 0.509, study coefficient we also got, that is a 0.46713, and we can do the analysis. The data is you can also get this data into the another sheet also. So you have to choose the option. So
in this way, in Excel, you can perform the whatever your problem, you can easily find out the regression coefficient. So now next is a correlation coefficient. So what is a correlation coefficient? So for example, if I'm asking the question, is there a relationship between A and B? So what is this A and B? A and B are nothing but it is a variables. Okay. So if I want to find out the relationship between A and B, then I have to use the correlation coefficient. Okay. So correlation is a relationship between the two variables, whether and in this case, mostly we consider X as a independent variables and Y is a dependent variable. So here X will be the independent variable and Y is considered as a dependent variable. So data is represented by a collection of ordered pairs. Okay, so collection will be like this, X and Y. Okay, so the linear relationship between two variables is represented by the correlation coefficient. Okay, so we have to find out is there is any linear relationship between two variables. Okay, whether they are correlated to each other, whether positively or negatively. Okay, so that correlation coefficient can be represented by the linear relationship between the two variables. So normal distribution of the data, if the data is normally distributed, well distributed, normally distributed, bell-shaped data is there, then at that times the correlation is called as a Pearson correlation, which is denoted by R. That is used for to understand the correlation coefficient. So Pearson's R correlation is calculated by using this formula. Okay, so the formula is given here, x and y. These are the variables, and n is a number. So n is a number of observations. Here x, y are sum of the product of, that is a variable a, and b, y summation of x, that is a sum of x square, summation of y, sum of y square, and like that we can put all this value in this equation and we can find out the Pearson or correlation coefficient. So there are different types of the correlation. Positive or negative correlation is there, simple, partial or multiple correlation is there, linear or non-linear correlation is there. So there are different methods are also there to study the correlation. First one is a scatter diagram method, another one is a graphic method, and third one is a coefficient of correlation method. So there will be always number between -1 and +1. So if you want to find out the coefficient of correlation, the value you'll get either in a negative or it will be in the positive. If our R, that is a Pearson coefficient, if it is close to 1, +1, okay, positive towards the positive, then we can say that all the variables are the positively correlated. So here you can see that there is a x and y, for example, okay, plotted, and this is the data is plotted. Now here you can see that for example, this is y and this is x. As I am increasing x, y is also increasing. So that means if I am increasing x, y variable is also increasing. So relationship is linear, and the value is towards the positive. That means either if it is a 0.9, that means it is close to the 1 positive. So such kinds of the relationship is called as a positive correlationship. In another case, if the R is close to -1, for example, if I am increasing x, if I am increasing the y and x is a decreasing, here you can see that the value of x is a decreasing as I increasing the value. So either one, either you are increasing x or the another value will be decrease. So such kinds of the relation. For example, here you can see that the value of x will be top, okay, highest value, but it will be reduce as the concentration of x is increasing. The value of y will be reduce and go down. That means x is affecting on the y value, and automatically the negative relationship will be there. So the whatever the R value you will get, that will be close to -1. Such kinds of the slope or such kinds of the relationship, it is called as a negative correlation. For example, effect of antibiotics on the growth of microorganism. In this case, what happen, if you are increasing the concentration of antibiotic here, automatically the growth of microorganism will be reduce, decrease. So that means there is a negative correlation. If our R value is close to 0, that means these variables are not correlated. There is no any relationship between. Either you increase x, doesn't going to affect on y, or you increase y, doesn't going to affect on the x. So such types of the data, here you can see that it is a scattered data. There is no any pattern. Here you can see that the linear, but it is a negative. In previous one, it is a increasing, so it is a positive, and in this case pattern is a decreasing, so it is a negative value. But here no linear relationship, either no negative, no positive, nothing is there. So that's why it is a and you can say that there is a no correlation, because R value is close to 0. So if here you can see that if there is a strong linear line is there and all the data is very much close to this line, that means it is a strong positive. Okay, so it is a increasing manner. So that's why it is called as a strong positive. If the data is not well fitted, I mean little bit there is a they are away from the main line or this linear line, then data that this is called as a weak positive correlation. Strong negative correlation. Similarly, if the data one factor is affecting on another and the pattern is like this and all these points are very close to this line, that means it's just strong negative correlation. Weak negative correlation, that means little bit away, points are little bit away, so it is a weak. And moderate, that means it is not very close and not very far away, so in between. So moderate negative correlation is there. And if the data is either in not decreasing manner or not increasing manner, it is a not well I mean there is a no normal distribution, anywhere data is scattered, so no correlation is there. That means X factor is not affecting on Y and Y is not affecting on the X, vice versa. So like this. So making statistical interference from the Pearson. So for example, you carried out the Pearson coefficient and you got the value. But again, the question comes in the mind that whether whatever the you got this correlation is because of it is by chance or it is really true, true of the population. So such kinds of the doubt will be there in the mind of the reviewer when you send any kind of the research paper. Why paper are rejected? Paper are rejected because most of the people, they don't know how to do how to use statistics in their research paper. So most of the, I saw that I myself, I am a reviewer for the, I checked many international research paper. Most of the papers are rejected because of lack of scientific, I mean statistical analysis. Due to the lacuna of statistical analysis, most of the paper, 70 to 80% paper have been rejected. Okay, now you understood how much it is important, how the statistic is important in the research as well as to publish the good research paper. So if there is a doubt in the mind of the reviewer will say that do the further analysis. Then how to do the further analysis? So you have to find out whether the your data which is obtained true or not, or whether it is a by chance. Whatever the you got the result, it is due to the by chance. Then you have to go through the two hypotheses. That is a null hypothesis. There is a not a significant correlation between two variables. Any observed trend or relationship may be due to the chance. Okay, again, if you want know any trend, positive or negative, so that means whatever the you got the trend, it is because of the your chance or maybe because of the sampling error. So due to our sampling error or due to the chance, whatever the result you got, so null hypothesis will tell you that this is a problem. Sampling error will be there. Alternative hypothesis will tell that there is a significant correlation between the two variables. So observed trend or the relationship between relationship is most likely not due to the chance or sampling error. Okay, so it is not due to the chance or it is not because of the sampling error, it's a true value. So therefore, these two hypotheses are valuable. We'll see one example. In this example, the time X in year that the employees spend at a company and the employees hourly pay Y for the 5 employees are listed in the table below. Calculate and interpret the correlation coefficient. Here the employees are given and then we have to find out their hourly pay. Their hourly pay in the form of the Y and time X. So if they are spend more time, they will get the more pay. We have to find out if there is any correlation between their payment and their time spent in the company. So we have to find out. So again, you have to use the same formula, that is a Pearson coefficient. So X is already given in the table, Y is already given. So you have to take the summation of X, summation of Y, X square, Y square value and XY value we have to calculate. You have to take their summations and then put all these value in this equation. And finally, you'll get the value. So whatever the value you got, that is a 0.97. Okay, our value is got the 97. Now, how to interpret the result? Most of the time you are doing the analysis, but you don't know how to interpret. What should I say next if I'm getting the value 0.97 of the R, Pearson coefficient? So that means 0.97%, this value is close to our 1. Okay, so R is close to 1. That means it is a positive correlation ship. So if you spend more time in the company, you'll get the more salary. That means salary is basis on the R's. So our R value is close to 1. So there is a positive correlation. If you spend more time in the company, you'll get the more pay. Similarly, in another equations here, you can see another example. The table is given number of absence, that is a X, and in calculus course, and find and the final exam grade Y. Okay, final exam grade as well as the number of absent students are there for 7 students. Find the correlation coefficient and interpret your result. Okay, so here X variables is given, Y variables is given. Again, put the all values. We have to calculate the all values, and that is these are values, and then put in the equation. You'll get the R value. So if the R value is minus 0.93, that means this value is close to our minus 1. Okay, so that means there is a negative correlation. So what is a negative correlation? So this indicate that there is a strong negative correlation between number of absence and their final exam grade. That means if the students are absent regularly, so in their exam they could not perform well, so their score is very less. Okay, so this was about the negative correlation. Okay, so that means absentee is giving you negative marks or less mark. So this is a negative correlation. If you are absent, you will get the less mark. So more absentee, more less mark. Then third example, here also we can see that the height, that is a x in inches, and the pulse rate. So a pulse rate, that is a y per minute for 9 people, and find out the correlation and interpret. So we have to find out there is a relationship between height and the pulse rate of the person, that is x and y. So data is already given. So we have to find out, we have to find out all the values. And once you calculate all the values, you have to put in this equation, and then you will get the here R value. Now here you can see that R value is minus 0.15. Now what will be the our interpretation? So in this case, if you observe that the 0.15 value, negative value, this is not a negative. I mean, this is not strong negative. It is mostly close to 0. Okay. So here you can see that it is a mostly close to the 0, and this indicate that because minus value is not strong, not 0.85 or 0.95, not more than 0.85. So that is the reason now it is very close to the 0. That's why it is found that there is no correlation between the height of the person and the pulse rate. So this was about the correlation coefficient example. So I will show you. So I will show you one video how to find out correlation coefficient in Excel also. So we'll take the
this example and how to solve this example with the help of Excel. We'll see right now. So just you have to open the Excel sheet, and on that Excel sheet, the X variable and Y variable, you have to type the values of the X variable, and you have to type the value of the Y variables. Now we want to find out if there is any correlation between X and Y variables. So what we have to do, so we have to click on the data analysis, and then in data analysis, there are different options like then click on the correlation, say OK, then output, you want to output on the sheet only, select the sheet, then you have to check the click on this range, let's say levels, and just say OK. So here you can see that this is a X and Y, and this is a correlation. Okay, it is about 0.96. So this is very close to 1. That's why there is a strong correlation between X and Y.
So now you understood how to do in Excel also. So how this correlation is useful. So mostly this is useful in biological research. Correlation analysis used to understand the relationship between the independent variables with the dependent variables. Independent variables such as the risk factors, for example, risk factors, and dependent variable, that is a disease outcome. Okay, so what is a risk factor and disease outcome? That is a dependent variable. So we can find out such kinds of, is there is any correlation. For example, if the 100 people are exposed to the market, so that is a risk factor, exposure. Okay, exposed in the market, and how many people got the corona, that is a disease outcome, that is a dependent variable. Okay, so the people, those have the corona, they are a dependent variable, and those who are exposed, they are on the risk factor. Okay, so they are independent variables. Okay, so for example, 1,000 people are exposed to the TB, but not all 1,000 people will not affected. That's why they are called as an independent variable. Only there is a risk factor. Okay, chances are there, but dependent variables, that means those who are the susceptible, and automatically they got the disease. Okay, so out of 100, if the 10 people are infected, that means it is a disease outcome. That is a dependent variable. Now we have to give treatment to these 10 people, but there was a risk factor, that is a dependent variable. So these kinds of the study we can do very well with the help of the correlation. We can also find out the what is a correlationship between the microorganism and plant growth. What is a correlationship between the your salary as well as the interest rate or your tax. Such kinds of the, is there is any correlation between your payment and the tax, that can also be find out. That means it is useful in the different field, in botany, in zoology, in microbiology, in commerce and arts. So all these statistical tools are useful to all the streams. So again, the same thing, if you want to do further check whether your data is correct or not. So correlation coefficient, it is significant or not. And that with the help of the t-test, you can use the t-test, and we can also do the further analysis. And automatically, after that analysis, your paper will be accepted in the good journal or in the reputed journal. So to publish in the good reputed journal, another important thing is that your way of writing, your way of presentation, your way of statistical analysis, and your way of graph, as well as the error bars, all these things are the valuable tools. So in biology, we'll see the why the error bars are so important in the graph. So most of the times when you do PhD, or you must be registered to the PhD, or you are you must be communicating some research paper to the good journals. So at that time, when your paper will be reviewed, reviewer will see whether there is error bar or not in your graph. If there is no error bar, your paper can be rejected, or it can become for the revision also, depending upon the situation, or depending upon what the reviewer is thinking. So in biology, error are most often represent the standard deviation of the data. For example, if you got the data, for example, I am saying, I want to check my glucose level in of my blood. Okay, so my blood is a population. So whole, for example, I have 5.5 liter blood. I will not take all the 5.5 liter blood to for the analysis of the glucose or RBC or WBC. So to check the quantity of glucose in my blood, I'll take hardly 1 ml or 0.5 ml blood. So that 5 ml 1 ml blood or 0.5 ml blood is a sample. Okay, so it is a sample, and my whole blood is a population. Now, if I took 2-3 samples 2 or 3 times, and whatever the data I got, and I got the variation in the data little bit. Okay, so how much my samples? Okay, 3 times I took the sample, I will take the average. Okay, for example, it is a 20 mg, and in every time samples I'm getting the 10, 12 or 13. Then I want to find out that whatever I have did the sample, from that sample, how much original value is there. For example, original value is a 100 mg, and in my sample I am getting 60 mg. So there is a 40%, I mean there is a 60 mg variation is there, deviated is there. So this deviation is shown by the error bar. And if my data is very much deviated from the original one, for example, in my blood, original glucose is, for example, 180. And if I am showing 300, so that means it is wrong. If I am showing only 20, that means it is wrong. For example, if you are going to the market and if you are giving the 100 rupees to the that person, and you are taking duster of 20 rupees, and that person should give you 80 rupees. But if the person is giving only 10 rupees, so what will happen? There is a deviation, variation. Okay, so that is the reason. That means you will not accept 10 rupees. you can accept 80 rupees only, or sometimes you can accept 79 rupees. 1 rupees is not there. You'll say that okay, it is negligible, I will accept it. Okay, so such kinds of the deviation is we can accept it. But if there is a more variation, more change, more your data is deviated, that is not be accepted, and that's why error bars is there. So here you can see that here error bar is a little bit more, here is little bit more here, but here is a less, here is a less, here is a less, medium. So like that, this shows that your main value is here and this much value is deviated, either plus or minus, either plus or minus. So that is acceptable. Then your paper will be accepted. So that means why to include error bars in the graph? Because we have to find out how your data, how much your data is deviated or spread around the mean value. For example, if mean value is 20 and your data is 19, that means your value is close. For example, your mean value is 20 and your data is 1. That means it is very away. That means your data is not authentic. Your paper will be rejected. So the reliability of the mean value as a representative number of the data data set, that is more important. For example, if I am not diabetic, but because of your value, the person will be declared as a diabetic, because you are showing the error there. Your analysis is not good, and because of that your analysis is not good in the laboratory, you are showing more sugar to that person. Person is not diabetic, but result is a diabetic. So that means it is a very much horrible or problematic in research paper also and in medical science also, and that is the reason these error bars are very much important. And that's why when you go to check for your blood, uh, the instrument automatically they will take the 3-4 times samples and automatically they will calculate and they will take the mean and they will do the statistical analysis with the help of the software which is attached to the that particular machine. So nowadays all the machines are now statistically well equipped, and that is the reason there is no chances of the error, and that's why statistic is so much important. Now what do error bars indicate about the that is the statistical significance? In any experiment or observation that involves sampling, if you are doing the sampling, at that time it is very important. If you want to check the cholesterol level in the blood, so that means you are taking the sample of the blood, or your whole population, that means 5.5 liter blood will be the whole population. From that you are taking the sample. So there is a chances of error. Where will be the chances of error? When the person is taking your blood, if he is not taking properly and if it is not doing properly analysis in the laboratory, if he is adding water in that, it is diluting, automatically your result will be faulty and there will be the error, or sometime due to the chance you are getting the results, and that's why error bars are so important. When we do analysis, at that time we'll get the 3 patterns. Okay, so here you can see that if your error bars are overlap, okay, so overlap, that means they are overlapping, that means this difference. Okay, so this here you can see that there is no significant difference between these two samples. Okay, so this is population for example and this is a sample, so value is very close, so this much error will be there. Okay, that error can be shown in your graph. So when standard deviation error bars are overlap quite a bit, it's clue that the difference is not statistically significant. This show that this is not that much significant. For example, if you are giving the 100 rupees, you should get 80 rupees back, but they are giving you 78 or 79, but you are accepting it because the value is not that much significant. In another case, here you can see that there is a less overlapping. Okay, here you can see that population is there and this is sample and you are getting this value. At that time when the standard deviation errors bars are overlap even less, if there is a less overlap, then what you this is clue that difference is probably not statistically significant. You must actually perform a statistical test to draw a conclusion. Then reviewer will be tell you that please perform another statistical analysis, or reviewer will also tell you that again do the same experiment for the 2-3 times and again do the statistical analysis to reach your conclusion. If there is a no, here you can see that there is no overlapping. That means here you can see that there is a significant difference in the standard deviation error bars. So no overlapping is there. So this is a clue that the difference may be significant, but you cannot be sure. This difference, you cannot surely say that there is a no statistical significance. So for that, again, you have to do the significant statistical test, or you have to repeat the your experimental data, and again you have to prove it. When such a two situations are there, again you have to do the experiment, or again you have to do the statistical analysis further. If you are not getting that much error, that means your data is okay. Reviewer will not take action against you. If the data is not up to the mark, at that time reviewer will tell you for the revision. So how to draw all these bar graphs in the Excel? So here uh, simply you have to insert uh, insert all these error bars in the Excel. You have to draw the first whatever the data you have, you have to draw the graph, and in this graph then you can click on this, and then you can find out there is a more option titles, and from that uh, there will be the standard errors, error bars also there. Click on that, and then more error bar options, click on that, and again you have to calculate the SD, that is the standard deviation. This will be your mean value, and this will be the standard deviation. Then standard deviation, whatever the standard deviation you got it, then you have to uh, put all these error values, and then you'll get the error bar on this graph. Okay, then you can also find out the style, what kind of the style, both. So click on the both, and you then specify the path, and automatically you will get the error bars in the graph. So how to do in Excel, I will show you the demo.
Dear friends, now you understood the importance of error bar in the graph. So how to add error bar in the graph in Excel? Trial code, and these are the values, and this is a mean of these values, and the standard deviation you have to calculate. So once you have the standard deviation here for trial 2, for trial 3, and this is for trial 4. Now what you have to do, you have to draw the graph. Okay, so I have drawn this graph. You have many, you can do so many changes in this graph. You can do title, you also plot on y-axis, x-axis. So depending upon your requirement, you can draw the graph. After drawing this graph, then click on this graph. Then after clicking like this, design structure will come, and at the left side there is the add chart element. Click on that, so you will get the sustenance of the then click on error bar. So just click twice on this graph, and at the left you will find the add chart element. Then click on that, and then click on error bars, and from the error bars you will get this much options. So click on the more error bars options. Click on this graph, and then at the left you'll find the add chart element. Click on that, and then find the error bars. From that, click on more error bars options. After clicking on that, series 1. So okay. So for the series 1, now at your right side, click on this. Now you have to choose the error bar, whether the both or only minus or only plus. So depending upon your choice, the generally people use both in research paper or the graph. style, whether no cap or you want cap. So you have to click on the cap. Then from these options, fixed value, percentage, standard deviation, standard errors, here we have to click on the custom. Okay, because now you have calculated the standard deviation and just click on specify value, and here positive error values. So how much error is there? So how much standard deviation is there? So click on that. For series 1 it is 7. So again for negative value also you have to take the 7 only. Okay, and then you have to say okay. So here you can see that for series 1 now there is a error bar on the graph. So in similar way now we can also
Sir, actually, voice is not clear. Could you repeat it again? Hello?
Yes, sir.
Voice is not clear. So could you repeat it again?
Okay. Just a minute.
From the beginning, actually.
Hello, can you hear me right now?
Yes, sir. Yes, sir.
Can you hear me?
Yes, sir. Yes, sir.
Simply you have to plot the data, which is shown here, and you have to plot the graph. Okay. And once you draw this graph, there is a mean value you have to calculate. Then standard deviation you can calculate from this, and you can then label this title. You can also label whatever the data you have, and then you have to add the error bars. Okay, so to addition for the addition of the error bars, then you have to click on this add chart element. Okay, and then from that you have to add on the error bars. Okay, so once you click on this error bars, you will get the other options. Okay, so what kind of the other options are there? So after clicking again, you have to click on this graph. Then once you click on this graph, there will be the add chart element. Okay, I am again telling you, add chart element, click on that, you will get here error bars. Okay, after clicking on the error bars, then just click on the more error bars options. After clicking on that series 1, then say okay. Okay, and then after that, then at this side you can see that the at the right side. Okay, so at your right side, once you do the this process, at your right side there will be the options. Okay, and at the right side click on this icon. Okay, after clicking on this icon, then you will get the such kinds of the captions. Okay, so either you want to show positive as well as the negative error bars, so click on the both. Okay, so most of the research paper or the people, they are using the both. Okay, in some cases people shows minus or plus sign only, but most of the people shows the both plus and the minus, so click on the both. So after clicking on the both, then you can again at the below you can see the cap, whether the you have to use like cap like structure in your error bars or not. After taking that, then click on the customs. Okay, after clicking on the custom, then specify values. That means we have to take the value of standard deviation. Here for the series 1, standard deviation value is a 7. Okay, so 7 will be the standard deviation value, and that click on that. Okay, for the positive error value, And for the negative error value also, you can take the 7 also. So positive as well as the negative, both value can be taken. So once you say okay, so in the series 1 here you can see that the positive as well as the negative cap, okay, in the series 1. In similar way also, you can do the same thing for the other series also. Same thing you have to do for the series 2. Click on that, and after clicking on that, again click on this icon. Okay, you'll get both, and again do the custom, then specify value. Again, in next time we'll take the this 14.6. Again, for the both value, we have to take the 14.6 positive as well as the negative error value also. And in again second series also, you will get the error bar. Say okay. Now here you got the error bar. Okay, these error bars are big as compared to the previous one. Why? Because value is standard deviation is more. Similarly, for the series 3 and series 4, you can put all these values. Okay, in similar way for series, for example, 3, and we have to take the values. Again, click on this icon both and the custom. Click on the custom, then specify value, and then the put your value, that is 6.3, and again click on this, detect the negative value 6.3, and again say okay, and you will get the error bar. And similarly for the series 4 also, you can do the same thing. Click on this chart, and once you click on chart, add chart element, then error bars, then more error bars, series 4, okay, and then again click on this icon, and you'll get the then custom, again specify value, and click on this, again take the 15.2, again click on this, take the negative values, again take, and in this way all the your graph will get the error bars. Okay, so depending upon standard deviation, how much your value is deviated from the original value. Okay, so mean value or true value, how much it is deviated, that can be studied. So in this way, we can study, we can add the error bar. If you have any doubt or any problem, you can ask me questions. Thank you very much for your patience. And
Thank you, sir. I think you gave a lot of insight on the topic regression coefficient, error bars in graph for figures, and Pearson's method for defining correlation coefficient. Thank you so much for sharing your presentation. I know there will be some technical glitches, but it's okay. We can wait towards it and we can finish it off. Now, proceeding towards the question answer session, I would now invite Dr. Sheetal Banerjee to take over the question answer session. Over to you, Sheetal Banerjee.
Thank you, sir. It was a very nicely explained, informative talk full of the relevant examples in all the arenas. Perfection is not attainable, but if we chase perfection, we can tend.
Hello, Sheetal ma'am, you're not audible. You can take over, Pratima ma'am. I think Sheetal ma'am is not audible here. You can
Okay,
ask.
ma'am. Thank you for this opportunity. Numbers are the highest degree of knowledge, and it is knowledge itself. So we are acquiring this knowledge for the last 5 days, and hope all of you are enjoying. The first question which has been asked to sir is for regression analysis, need to select level of significance in Excel for better estimation of regression coefficient. I'll repeat the question. For regression analysis, is it needed to select the level of significance in Excel for the better estimation of regression coefficient?
very nice question. Yes, it is a necessary because as I said, when we do anything, sampling or anything, and we are going to find out any correlation between variable, either for example, I am finding new antibiotic, and I want to check whether my antibiotic is affecting on the particular bacteria. Okay, but now I am trying to kill that bacteria with the help of my antibiotic. When I am taking the trial with the different concentrations, and I am getting the MIC, that means minimum inhibitory concentration. At that concentration, bacteria kills or inhibit. So once I take that value, and I'm predicting that this is a MIC, this is the concentration, that is a 10 microgram per liter. This is concentration. At that concentration, bacteria will get killed. Okay, so I am predicting it, but I need to prove it statistically. Whatever I am saying that I have to prove. Then how to prove? I have to do it repeatedly, 3, 4, 5 times, and then I have to do the statistic analysis. I'm only doing the regression analysis. Then level of significance. Okay, that is also important. That will show that how much error free is our data. So significance is always important because it gives the more value to your original value. It make close to your original value. That's why significance of the data is very, very important.
Thank you. So nice explanation, sir. There is one more question. Which one is good for interpreting result based on R1 and adjusted R2?
Which one is?
Good for interpreting results based on R2 and adjusted R2.
So R1 and R2. I mean, this question is a little bit confusing about what I mean. With what
So
relation?
R2 and adjusted R2.
Okay, R2 and adjusted R2. So can you repeat again the same question, madam?
Yes, sir. Which one is good for interpreting results based on R2 and adjusted R2?
Based on R2 and adjusted. So sometimes adjusted value is required in some cases. Okay. So most of the times when some particular condition is there and you need to adjust the value. So most of the people or most of the reviewer or most of the editor, they give much more value for the adjusting R2 values, because at some condition it is necessary. Not always.
Okay, thank you, sir. There's one. Okay, sir.
Not always, but
there's
sometimes.
One more question. Yes, sir. Yes, sir. You can continue, sir.
Yes, I mean, so when it is needed, at that time it is we have to adjust it.
Okay, sir. Okay, sir. Sorry to interrupt you, sir.
Okay,
There's
no
one
problem.
more question from Falguni Patil, and she's asking, is it necessary that error bars are mandatory for good publication?
Very nice question, and this question is very important for any researcher, for students, as well as for teacher. These error bars are extremely useful and extremely necessary for any good journal. If the journal is not good, or if the journal is a predatory, they will take your data for the money making. I'm very frankly speaking, such kinds of the journal are not useful. The journal, those journal, they have standard good quality, like Springer are there, Elsewhere is there, Blackwell is there, Wiley is there. All these reputed with high impact factor, and they are also assessed by the Thomson router. Okay, this is the scientific agency. So such kinds of the journal, they will never accept your data without standard error. So that means you did not perform your experiment 3 times or 4 times. Okay, this indicates that your data is by luck or by chance, and your data can be easily rejected without any comments. That's why it is highly important. It is necessary. So if you want good publication, if you want to be good researcher, good academician, so error bars are very much important. You have to find out the standard deviation and then put the error bar, and that is very much valuable. I'm telling my own experience. When I sent my paper without error bar, when I was doing my PhD, my paper was again came back, and comments was comments was there of reviewer that there is a lack of statistical analysis, and that is the reason you need to do the major revision. And for 1 month I was doing the statistical analysis, And the very bad part is that these statistical people don't help to the biology people. And that is reason biology people or any other people, they are not from the statistical background. They can learn so many things with the help of the software as well as the by studying the books. Myself at that time, I decided that I will study the biostatistics very well, because that is very necessary for the research. And then myself, I did all these things very sincerely. I understood basic things of the biostatistics, and then I put all the error bars. I do the ANOVA, I do the t-test, and my paper was accepted in Journal of Hazardous Material. Now the impact factor is more than 6. So this is the impact of the statistical analysis.
Definitely, your inspiring words will inspire all our audience, and definitely they'll add the statistical part to their papers. And it was very inspiring to listen from you. And over to Alia Naz ma'am. Thank you,
Yes,
sir.
thank you, Pratima ma'am, and thank you, sir. Very well said. Statistics we don't consider or don't wave so much in other fields. And now, after you gave us this insight, I think we all should take serious note of statistics and to use all these things in our papers when we are presenting to the journals. Thank you so much, sir. Now, moving to our second session. And before we start our second session, I would like to give some notifications here. As in, MCQs of 50 marks test will be conducted on 8th of November 2020, where the link will be opened at sharp 7 p.m. and it will be disabled again by sharp 8 p.m. So I request all the participants to make sure that they attempt the MCQs for their certifications, definitely. And whatever updates regarding the FDP is posted on our website at www.theijmspublic.com. And second thing is the participants are going to get the national award for the best 3 participants. Again, after the FDP, when they attempt the questions, the results will be posted after 15 days on the website. So please keep in touch and stay tuned to the updates with our website. Now, proceeding further, we will start with a second technical session, which is on the topic contribution to educational innovation and patents. And patents are very important. So we have our honorable speaker here, Dr. S.R. Bamne sir, professor in chemistry and principal of Sushila Shankarrao Gadhave Mahavidyalaya, Khandala, Satara district. So here I welcome you, sir, for this FTP. And before we proceed, I would like Meenal Majati to give the profile of our speaker to the participants. Over to you, Meenal.
Thank you, Naz ma'am. A very good evening to everyone present here. I'm here to introduce of our today's session, Dr. S.R. Bamne sir. Sir is designated as Principal of Khandala Shikshan Samiti, Sushila Shankarrao Gadhave Mahavidyalaya, Khandala, District Satara. Also designated as Principal of Dr. Patangrao Kadam Mahavidyalaya Sangli from 2006 to 2013. The field of interest includes synthesis and development of bioceramics, gas sensing materials and bioactive materials, catalysis and photocatalysis. Dr. Bamne sir has been Senate member of Shivaji University, Kolhapur, September 2017. Reader in Chemistry, Shivaji University, Kolhapur, 2004 to 6. Member of BOS Chemistry in the same university from 2010 to 15. Sir has a teaching experience of long 34 years and research experience of 28 years with 4 patents on his names. 8 PhD students are working under the guidance of Dr. Bamne sir, while 4 for MPhil. The major research projects titled Studies on Synthesis and Development of Bioceramics and Compatibility, Testing Towards Nano-Enabled Drugs. Minor research projects titled Synthesis and Characterization of Gas Sensing System, and many more. Some of the programs under the guidelines as the principal by Dr. Bamne sir are started research laboratories in college for MPhil and PhD, implementation of UGC Network Resource Center, organized 9 national conference funded by UGC, work for member of selection committee for the post of principal, assistant principal, VC nominee. Apart from all this, Dr. Bamne sir has huge contribution in social work. Sir has been member, Giants Group Sangli, Indian Association of Chemistry Teachers, IACT, University of Lucknow, District Advisory Committee, Nehru Ministry of Sports and Youth Affairs, New Delhi, Director, Rajaram Bapu Patil, Nagari Pata Sanstha, Sangli. Sir has organized about 20 blood donation camps. Now I request Dr. Bamne sir to take over the session. All over to you, sir. Thank you.
Thank you, Mitali. Good evening to all and welcome to my talk. I'm sharing here with screensaver. Gote, Choto. Screen.
I think we lost the connection of Bamne sir. Hello, sir.
Hello, I'm sharing my screen.
Yes, sir. If you want help to share your screen, can I share the screen for the presentation, sir?
Wait 1 minute. I will try, then I take your help.
Okay, sir.
Desktop.
I request all the participants to stay tuned along with us. Anytime now, we will be starting the session. Sir is about to start with his presentation. And I request all the participants to be updated. Yeah, here we go. We are going to start now.
Ma'am, I'm audible and visible.
Yes, sir. You are audible as well as visible, sir. You can start, sir.
Welcome to this 1-week faculty development program on statistical analysis in academics and research. My topic is contribution to educational innovation and patent. This very nice faculty development program organized by International Journal of Microbial Science in collaboration with Sushila, Shankarrao, Gadhave, Mahavidyalaya, Khandala. So I will discuss about this slide later on. My API and patent review. Why I will show this slide to you. My API score calculated on the basis of UGC notification. First notification date 2010 is 1,300. And after fourth amendment, I have calculated my API for 540, and last UGC's amendment on API calculation is on 2008. It is 538. The 538 API is calculated. I have published more than 57 research papers, which have impact factor more than 10, and it is calculated from Thomson and Reuters method. I also rightly predicted tell that I in 2 patents in my credit and also 2 patents are published. Innovation. This is the era of decade of Indian innovation, 2010 to 2020. Almost we are reaching towards the end of the innovation decade. India's global ranking of innovation in India is good. Innovation refers to doing well. So this is a traditional story of thirsty crows. The innovation means it is a technology plus creativity. Innovation means the education. Innovation plays an important role in globally developing skilled force. As a result, innovation in education is very simple, not teaching blackboard and books, but it can be teach by new methods. I will give a recent example, that is Dongar Shala, Sugar Shala. In today's, we have also observed that we have no connectivity, no internet connectivity. For that purpose, some teachers go to the hillside, step on trees, reach on the mobile tower and collect the internet data and come down and teach to the students. So this is the innovation in teaching. In the same way, some teachers go to the slum area or sugarcane harvesting camp and teach their kids. So this is also innovation. In many decades, we are teaching with textbook and the blackboard. So we have to expect the new style of teaching, and this new style of teaching, that's technological innovation, having the significant impact in the educational system. Online courses, teaching aids, educational softwares, social networks tools, and others emerging technology are disturbing the traditional classroom development. In this modern educational system, major area of the data analysis and cyber security of students are mainly concerned with. So we also teach the computer assisted instruction, website, and social media, laptop, and e-books. About the invention or education, a DNA inventor, writer, Diner, Gerson, and Christensen rightly said that the whole world has a question, and the inventors identified the 5 characteristics common in great innovative entrepreneurs. These are the questions. The innovators asking the questions, some observations, experimentation, association, and conducting of dots and networking. So, some organizations such as IITs, universities, national laboratories plays a very important role in the innovation in educational system. They must spread the awareness regarding the Intellectual Property Act, and from 2008, most of universities have intellectual property right development cell. They have introduced and they are spreading the awareness regarding about the innovation and the patents. The organization must continuously, perpetually focus on the awareness-based grades. They helps the reverse these negative trends by investing some new trends, sophisticated equipments, other equipments such are useful for the new researcher. the organization must recognize their research value. They recognize their work. They appreciate their work, and the organization should organize the training on how to filing the patents. Then what is patent? Patent is an exclusive right granted for an inventor. According to Patent Act, Section 2, invention must means a new product, new process involving in the interactive steps capable for the industrial application. The new means, I will give the classical example. The new means the research innovation should not be published earlier. Patents should not be published earlier. So it is called new. I will give the one example. That is patent filed by US Amuri. Turmeric, neem, and basmati rice. Dr. Masha valiantly 14 months judicially fight and revoked this wrong US patent filed on the Indian herb turmeric. His efforts on revocation of patent would hailing the property of turmeric. Patent was wrongly granted to the United States. Patent on basmati rice granted to United States as rice cake. This is the long grain aromatic rice grows in India and some part of Pakistan. So this wrongly patented to the United States. So this is means new. So somewhere else it is not the file. So patent provides the protection for the invention of the owners to the patent. This is a statutory right of the inventor applicant given to the government. So patent has a limited life, 20 years only. I am audible? Yes?
Yes, sir, you are audible, sir.
I am audible?
Yes, sir, you are audible, sir.
Okay. Types of patent. So there are three types of patent. One is utility. One can use in a day-to-day life or our day requirement. Second, design, industrial building design, etc. Plant patent. So plant patents has a limitation. One cannot file a patent on plant grows with seeds. The patent can be filed on plant. Plant must be reproduced by asexual reproduction method and not by seed. So potted is also asexual reproduction method, but it has a limitation. It is what we can. It is a one. So one cannot file the patent on potato. So and to patent file on plant, it cannot be used for the public use. So these types of patent. Then role of patent system. What patent rights? So patent rights protect the inventors from third parties such as using, selling, marketing, and licensing. Encourage the inventors, promote the commercialization, accelerate the commercialization of invention and the whole society. What can all be patented? So invention research can be patented? No, just I have not said this. Planting must meet the several requirements to the eligible for the patent protection. Patented subject matter by any articles, any apparatus, machinery, or its component suitable for industrial application. It may be living or non-living. Some pharmaceutical products, any composition of matter used for manufacturing unit. What can not be patented? So these are the following list cannot be, one can patent, one can make research, but one cannot be patented this. So scientific theories cannot be patented, mathematical methods, plant and animal varieties. In India, Western Ghats, the animal kingdom, new animals invented, but one cannot be patented, such as a frog, one species of frog, butterflies, wall lizard, newly found in the Rajasthan area. So these cannot be patented. Plant and animal variety, discovery of natural substances, commercial methods for medical treatment. In medical treatment, we know that there are four blood groups: A, B, O, etc., etc. But India has found the new blood group. It is called Bombay blood group. So it cannot be patented. In prior to research are some literature, including the technical and scientific journal, cannot be patented. Textbook, conference proceeding, some website, these companies' website, brochure, some newspapers or newspaper trade applications, publications cannot be patented. Where to conduct the prior research? So once we do the research and we wish to patent this research, before patenting, we have to make some prior research, search out whether this patent or our research is in published somewhere other or not. Just like patent on turmeric patented by United States and it is revoked. So avoid this condition, avoid this situation, we should make a prior research, and this research patent and patent application published by many patent office. So go to their website, a list of IP, intellectual property office. They also give the free charges, the research on the some website also available to search or search research for prior application of the patent. Most national patent offices offer patent search services for free of charge. It is not easy to perform the high quality of patent search. Patent jargon is often complex and obscure, and professional searching requires the considerable knowledge and expertise. How to write the patent application. So essential ingredient of complete specification, we should give the specific title, preamble of your patent, and submit the form number 2, which has the complete specification, just like our research paper. Abstraction, then short story of your invention, like that. Then result and some patent. You have to search our research in the given website so that our invention should not be published elsewhere. Then its drawbacks, its merits, then invention. So write the very short what you have invented. Then statement of invention, giving a short title, then detailed description of invention. You have to write the how you invented, procedure, some methods, and these methods should not be published elsewhere earlier. Details description regarding the reference to drawback and claim. Claim should be not more than 3, otherwise it charges and it makes some complications. So ppm should be precise. For example, I have invented or my research used calcium silicate nano rods. So there are several applications of nano rods. Several scientists and inventors published papers, patented some article. So while searching this prior art, I come to know that nobody has invented calcium nanoride having size less than 100 nanometer. So we have searched this and then we have this size. That is our size nanoride size is 34 to 40 nanometer. Then we have also given another claim that we have invented method of preparation is very short. No earlier has been published. So so we can make a 3 or 4 maximum claims. If you add more and more claims, it may be complicated. We have to face the opposition and the questionnaire of the patent office. Main step for filing the patent procedure. First step, file the application to the or at the patent office. That is a form number 1. That patent rule 2000. Add all the details, address like this. Then filing the request examination. So once we file the details regarding patent, file patent online or offline, then we have to fill up one form to examine our patent with sufficient fees. In form number 2, we giving the complete specification. Then publication. We have to give them authority to publish our patent in the patent office publications, not in other journals. So then examination. Then examination for we have to fill up 18. Then pre-grantation. Form number 3, declaration that we have to again add the declaration that this innovation, the patent cannot be published elsewhere in India or outside the India. Then we have to fill up form number 5. The patent can be disclosed. So in my case, we have invented the calcium silicate nano rod having the nano size 34 to 40. So before this one, I also published or patented that he has invented the calcium silicate nano rod having size 100 nanometers. So this patent can be disclosed to us, that is refer this patent and give your answer at the time of examination. So you have to fill up the form number 5, can be our patent disclosed whenever necessary. Then patent is granted. And lastly, grant application. Like patents, it helps to gain the entry into the market and discourage our opponent. So this is the first step. We attract the inventor. Suppose we have invented a patent on a LED bulb or having a carburetor of a petrol car, use the more mileage. So then attract petrol manufacturing and then car manufacturing investor. So best be based on the marketing tool to promote the unique aspect of the product. Be used as a collateral to obtain the funding and increase in the profit power. Create a revenue. Once you can file, your revenue is created. You use a more asset, more money. We give the license for selling our patent like a property. So patents are form of property, can be add your value asset. And moreover, today why we gather together, it helps to increase the API score. Why I have shown at first slide, that is my API score this. The API score is increased by 0.01 because I have, and also this patent is used to my personal in my college grade A grade, obtained A grade in NAC accreditation because of our principal me having the patent. Who can apply for the patents? A researcher, those who are inventor can apply for patent. Inventor assigned to this work. Suppose inventor and he can assign to work in Indian friends, has the patent, obligatory assignments. Those who have legal rights, he can also file the patent, and person with a sufficient proper of priority interest. Those who have business purpose, business interest, he can also file patent on behalf of inventor. Word, more than lakhs of patents sanctioned, granted, more than lakhs of patents. So it is very difficult to give the examples of patent, but only few I will give here. The pen with scanner. If you write on your notebook, it directly see on your species screen. Then horse nail. We have seen the horse races. They use the more painful to the horse. So avoid these one pet inverter, patented a horse nail using the tractor tire and relief. This gives the relief to the horses from pain. As you know, the dialysis can be invented. There's still kidney dialysis invented by Niels Alwal and life-saving invention. As you know, we are using our Ford car and we are also using the seat belt. But unusual three-point seat belt invented by Niels Bohlson and patent granted to West Germany by the Volvo company car making. The motorized skeleton also patented. This is an example of this. Lastly, I will give the classical, as you know, seven wonders of the world. In the same way, eight wonders of the patent in the patent world. First wonder, as you know, light bulb. The electric light bulb in perhaps most of the famous patented inventor known in the human race, and it is patented by Thomas Alva Edison in the year 1872. However, at the same time in England, Joseph Swan received the patent in similar product. So there is crisis. So they two meet together and form a electric company, Edison and Swan United Electric Company, to solve this problem. Edison, Thomas Alva Edison has more than 1,049 patents, including photographs, Paso gramophone, record player, dictaphone. Second interesting patent, internal combustion engine. Today, or and the age of today, it is impossible to imagine how would we function without the car, buses, and trains. These vehicles operated through the internal combustion engine. The first internal combustion engine was patented in 1823 by Samuel Brown. Then four-stroke engine in 1973 by Nicholas Ford, and two-stroke engine was patented by Carl Benz in 1872. So this is the boon of for the moment. This is the definite boon for the moment. The third patent is telephone. Telephone industry is a great industry and significant industry today. The first telephone was patented and invented by Alexander Graham in 1876. Third, fourth example, that is a wonder of patent, is a computer. First modern computer to be invented and patented by electronic numeral investigation computer. The patent went to the John Smootsley and J. Presser Eckert in 1973. However, United States court invaded this patent over the claim that Anton Berry computer ABC developed the computer in the year 1940 come first. So the first computer in 1970 patent is invented. Take back. However, the Apple company commercially successfully developed the Apple II 1997. Bluetooth is another wonder. Despite of popularity of mobile phone having Wi-Fi connectivity, people still opt for the Bluetooth as an option for data transfer. Invented in 1994 by Job Hudson, Bluetooth technology allows nearby electronic device to connect using the low power, ultra high frequency waves. Today, Bluetooth is the almost every handset device for multimedia transfer and reception. Next great invented wonder of patent is Magleville. Among the more interesting patent innovations was the maglev or magnetic levitation used to propel the train. The first patent for maglev went to the Brookhaven National Laboratory, United States in 1968. Researcher James Powell and Gordon Dabby came first to concept of using magnets to propel the train. The another and very interesting patent is Fire Eye malware system, that is antivirus system. Computer viruses are banned to information technology industry as well as to detect such virus was Fire Eye malware protection, and this system patented a engineer, Pakistani American engineer Azhar Aziz in the March 2005. In the same way, we are using antivirus system, and it is invented by Maharashtrian fellow Sanjay Katkar, 1995. The Google page rank. This is another invention. Another famous example of patent Google page rank. Larry Page developed in 1998. It gets its name from his surname Page. It calculates the importance of page by counting the number and quality of link attached to it. Example from the across the world demonstrate the criticality of the patents in the world. Patents give the individual exclusive right to their inventors, and through these rights, you have a strong market position. This leads to the fueling the fire for innovation. Finally, the patent provides the positive image for your enterprise, which can be useful for building a business. Now, somebody asked, what is the charges? What is the cost for filing the patent? This is the official charges for filing the patent. One can file patent on e-filing also individually. For filing the first patent, it charges about 1,600 in e-filing, and in presence you will charges the 750. In the same, this is examination and all this I have produced. It cost more about the 10,000 rupees. If you own file, and there are some agencies, companies available to help the filing or patent, but they charges from 10,000, 30,000 to 75,000, depending on cost. Patent crucial for India to bridge the tech gap with United States and China. So India lags far behind global leader in the patent. Worldwide, 1.4 million patents filed in every year. But India, I will take the example in 2007, India files only 12,000 patents, while China files 4 lakh 20 patents. In case of US, it filed granted 3 lakh 18,028 patents. In the same way, Japan also more than 1 lakh patent granted and 2 lakh filed. But India is very small, 12,000 granted and 40,000 something filed. So India is very lag behind in globally filing the patent. So this is because I think it is not a critical position. I'm not criticized to any other, but it is because of lack of fund. India spent less than 0.7%, less than 3% of its GDP to research and development, while other countries, US, China, Japan, spends the more than 2 and 2.1% of GDP for the research and development. This is the one reason that we have lack of funds. Second reason is what I have faced is in India, patent granted takes the more officially takes the more than 2 months, while in other countries it takes the 15 days or maximum 2 weeks. In my case, I get patent in 1.5 years. So this is the why we get the less patent fund. In India, the universities, IITs, so they also file patent, but CSRI is the top among the Indian patent filing institutes. So in 2017, 2018 and 19, CSR granted the patent 166. Lastly, is very much like to me, that is then American social thinker, reformer, wrote that grant me serenity to accept the things and cannot be changed, the give courage to change the things I can, and the wisdom to know the difference. In Marathi, I can say, हे ईश्वरा, परमेश्वरा, ज्या गोष्टी आम्ही बदलू शकत नाही, त्या शांतपणे स्वीकारण्याची शक्ती दे. ज्या गोष्टी बदलायला हव्यात, त्या बदलण्याचं धैर्य दे. आणि वरील दोन्ही गोष्टी ओळखण्याची हुशारी दे. Thank you. If any question.
Yes, sir. Thank you so much, sir, for your insights on the kind of topic we had today. And it was really inspiring to know that so many things are happening and to guide us in the path of doing whatever we have to in the right direction. And that is patent rights, which many of us are not aware of it. And with your presentation, we came to know many facts about this concerned topic, which we were not aware of. Thank you for sharing your knowledge with all of us, sir. It was pleasure having you on board. Next, we are having question answer session. If any queries of the participants will be posted over here. So I hand over to Dr. Pratima Pawar ma'am to take over the question answer session. Over to you, Pratima ma'am.
Thank you, ma'am, for this opportunity. And the first question for our speaker is why only 20 years validity for the patent?
This is the
Why is it that there is only 20 years validity?
20 years. They have given one limit. That is, it has not a significant value to the number. They give only 20 and 20 years is a maximum. After 20 years, it is available to the common people.
Okay. Thank you, sir. So there's one more question. Can genetically modified microorganisms be patented? This is asked by Amrita Pandey, and she's asking if genetically modified organisms can be patented.
According to my list, it may be patented, but it comes under the 3 categories. Plant, the microorganism, is it a plant
No, sir.
Then it may be. So we will search more on the website or patent office. We can ask them whether if it is a really just like a corona, useful for corona or like this.
Which are the different patent
Continue.
authorities? So the next question is, which are the different patent authorities, patenting authorities?
Achha. Patent authorities in Maharashtra, Mumbai, Kolkata, Madras, United States patent. If you want to file the patent outside India, you can file outside. But there
So what
is just like a university, there's separate patent authority, intellectual property office.
okay, sir. So what is the difference between international and national patenting?
International and national patent. So the name itself suggests can be internationally. Patent file in international, it can be used in national country. And if you file in India patent, it is available only for India.
Okay, sir. Okay. There's one more question asked by Party Falguni, and the question is plant products such as Ayurvedic ointments, medicine, etc., from the plant extract. Can it be patented? The byproducts or plant products, can it be
Yes, it can be
patented?
patented, and its classical example. The Ayurvedic products can be patented. Its classical example is neem. So neem root or neem medicine patented by United States. It is revoked by India because Indian can use traditionally the use of this Ayurvedic plant. So one
Yes.
can patent.
Yes. Thank you, sir. There's one more question. What is the importance of patent in social science?
I know the social science students generally ask, it gives the value, it gives the status to the society using this patent. And if it is a commercial, then it will add your value asset.
Okay, thank you so much, sir, for your valuable insight into this topic of patenting. And you also have explained even the expenditures and the statistics related to the patenting process. So once again, thank you from the organizers. I thank you once again for your valuable and nice guidance. And it was a wonderful session listening from you. And over to Alia Naz, ma'am.
Thank you, Pratima ma'am. As we have come to the end of our session for the day 6, hoping to see you all for tomorrow, that is day 7 session. Before we wind up, I would like to give some of the details for tomorrow's session. Day, se- Day 7, we have session 1 on sample size calculation in research by Professor R.L. Shinde, Department of Statistics, KBC North, Maharashtra University, Jalgaon. And second session will be Teaching and Participatory and Innovative Learning by Subrota Hathi, Assistant Professor, Department Dairy Microbiology, SMC College of Dairy Science, Anand Agriculture University, Anand, Gujarat. Hoping to see you for tomorrow's session. Before we wind up, I would like to end with this quote. Life's not about expecting, hoping, and wishing. It's about doing, being, and becoming. So, hoping to see you tomorrow for the last day of our session. And I sincerely thank all the speakers for being so very good in their presentations and giving us and sharing their knowledge during this pandemic situation. Even though we are from different parts of the country, we have come together on this platform, which is organized by IJMS, that is International Journal of Microbial Science, in collaboration with Sushila, Shankarrao, Gadhave, Mahavidyalaya, Khandala. It gives an immense pleasure to wind up the session to me seeing this kind of response. I am really happy and hoping to see you all tomorrow. Stay home, stay safe, all of you. Bye for tonight.